DSM, the Diagnostic Statistical Manual, says that to have Tourette disorder, you need to have multiple motor, that means more than one motor tic and at least one vocal tic or phonic tic at some time during the bout of illness. And as you heard earlier, it waxes and wanes. It has to at least last a year because many kids will have a bout of a short period of time where they have tics and it doesn't go into a disorder because it's only a short time. It has to have an age of onset of under 18. This is our current definition. The definition probably will change again. And it's not due to some other effects of substance abuse or other medical conditions. That's how we currently define it. However, there are other tic disorders we don't talk a lot about in general, because Tourette is the big one that everybody talks about. So in particular, I want to focus on this other chronic tic disorders, either having motor only or vocal only. So you can have a chronic motor tic disorder or chronic vocal tic disorder. And you don't have the other, you don't have to have multiple, okay? And I want to point that out, and I want you to remember that one because it's a little, we're going to talk about the genetics and talk about that one. And then there's these others. One is called a provisional tic disorder, where a year hasn't gone by yet, and we don't know if they'll stay having these tics for a year. So it's sort of a tentative diagnosis. And then there's sort of the garbage pail term, other tic disorders, where that people just don't fit into. People don't always read the DSM, so they don't fit into our categories. So the epidemiology, the prevalence is between 0.3 and 2.8%. So that's pretty common, especially 2.8%. And it does vary by setting. If you're in a clinic versus the general population, is it the gender? Obviously, males higher than females. And if you include other chronic tic disorders, is one. And the word is other chronic tic disorders because Tourette is a chronic tic disorder. Okay, it's higher in youth than in adults. I'll talk about why a little bit. And it's higher in males and females, and we don't necessarily know why, but it may have a different manifestation in females. And it is worldwide. As far as we know, it is worldwide. So there's some myths. First, it was very rare. No, not really rare. It's relatively common. Is it very severe? No. We have a lot of people we see who are just very mild tics. It never, like for example, we'll see a kid who has very severe tics. Parent, says, "Oh, I've had this little tic all my life. It never bothered me that much." They have chronic motor tic or chronic vocal tic disorder, whatever. So it can be, it's variable. Some people very severe, some people mild, and severity changes over time. And then, of course, as you heard, coprolalia is rare, so 5 to 10%. So it's not really, even though it's the TV version of Tourette, it's not. It is uncommon. Okay. So it is associated with other disorders, in particular OCD, obsessive compulsive disorder, and about 50% of the people who have chronic tic disorders, I'm going to try to use those terms instead of Tourette, have OCD. And over at least 50% or higher have ADHD. And initially, they never even considered that autism was really involved. I talked to some of the people who did the original studies, and they excluded people with autism. Well, if you exclude them, you're never going to learn about them. So there's more recent evidence that there are some families in which there are people who have both Tourette's or chronic tic disorders and autism spectrum disorders. And I'm going to talk more of it, so remember those. I'm going to be coming back to those when we get into genetics. So course. Course is an interesting thing. There was a question earlier about course, why people get better, or some people get better. We don't know why some people get better. But in a study looking at people over time, about 37% self-report that their tics get better. They're not having tics anymore. And others have mild tics. And some people, and about 19% have worse, pretty bad tics for the rest of their life. We don't know that why. It would be great. While the tics may change, The associated conditions like OCD do not. So in some ways, they don't really go away. The disorder doesn't necessarily go away if you consider OCD as part of that. And then lastly, I want to mention, it's really important, that this self-report of 37% may be an overestimate, because that's a self-report. There was a study showing that you get some people together who said they no longer have tics, and they videotaped them as they were hanging around talking to people. They have tics. Or the wife will say, "You still ticking?" Even though he says he doesn't have them anymore. We also talk to people who say, you know, when I'm really under a lot of stress, I have that urge, that premonitory urge, and I still maybe feel a little tics. Maybe people don't notice it, but I still feel it when I'm under a lot of stress as adults. Okay, so now, a little background on that. Now I'm going to talk about the genetic epidemiology. So, interestingly, if you read the article, the original article by Tourette, he thought there was, even though the idea of genetics was not really well formed back then, he thought it was hereditary because a number of his patients, he noted that their siblings or relatives also had tics. So he was even thinking something about hereditary at the time. Then we went through sort of the dark ages of it's due to psychological factors, like they did in autism with refrigerator moms and all that other terrible stuff. This is clearly not true, okay? But in the 1960s and later, they started doing studies showing that is due to some hereditary causes. And what you need to do, these genetic epidemiological studies, which I'm going to talk about. So, genetic epidemiology is a series of kind of studies that give you evidence that it might be due to genetic factors. And there's a series of them. So the first one, is the disorder familial? Does it run in families? For that, you do what's called a family study, which I will talk about. Then, given that you've shown that it is familial, what proportion is due to genetic factors? And for those, you do these hereditary studies, and in particular, heritability studies, in particular, twin studies. There was adoption studies too, but they were very, very hard, very rare, and there's no adoption studies in Tourette. So twin studies, and you probably remember from your high school biologies or whatever, there were things called twin studies. So I'll be talking about that. And then once you have that kind of evidence, then you say, it's time to start looking at the genes, to find out where those gene or genes are, where are they in the genome. So you do the molecular studies. So let's talk about family studies. How do you determine if a disorder is familial? Well, basically what you do is you recruit a whole bunch of people with the disorder. I'm just going to use Tourette for the moment, or chronic tic disorders, and a bunch of people who don't. Bunch of people, let's say 100 of each, but usually more. You then look at the rates of that disorder in their first degree relatives, parents, siblings, children. If the rate in the relatives of the people with the disorder, Tourette, versus is higher than the rate in the general population or controls, it's familial. That's a family study, okay? Now, not every trait that is familial is genetic. Wealth, religion, culture, and somebody did a tongue-in-cheek study of going to medical school showing that that's a familial trait. Okay? Now, some doctors might say, yeah, that's genetic, but I don't think so. Okay? So, not all things that are genetic are familial. I'm not familial are genetic. Okay, so in pictorially sense, you've got a bunch of people with the disorder, a bunch of people without the disorder, and you look at their relatives and you say, is it higher in the relatives of these people versus relatives of those people? Of those people. Okay? That's a family study. So, what do we know? Oh, I'm sorry, before I go into that. These family studies also can give you some idea on recurrence risk, meaning once somebody has it in the family, what's the chance that somebody else might get it in the family? And it's an average over many families. It's not as specific about a family that's in front of you, okay? And it's very helpful when there's no genetic test that is available. You can at least give some kind of that information to people who are interested, okay? And these are used by healthcare providers like a genetic counselor, which I was originally trained as, to help families decide and to think about what the risks are, okay? But there are limitations. There is no DNA being looked at. There's nothing about that family that's in front of you. It's an average over all different families, okay? So what do we know about the family studies? Many family studies, different populations, different countries, all consistently show that Tourette strongly aggregates in families, much higher in relatives of cases compared to relatives of controls. And if you include the other chronic tic disorders I was mentioning as being affected, the rate even goes higher. It's higher in cases. So a person has Tourette's and their relatives have other chronic tic disorders compared to controls. Yes?
Have you looked at ADHD and OCD in additional to the Tourette's?
Getting there. You might want to write questions down, so I'm trying to leave some time at the end, okay, so we can do it. But you can see there's a lot of studies here. So for one example, a very large study, looking at Tourette's plus other chronic tic disorder, there's an 18 times higher rate of relatives, first-degree relatives compared to relatives of controls, okay? And if you look at the second-degree, so that's first degree is parents, siblings, children. If you look at second-degree relatives, that would be grandparents, aunts, and uncles, okay? It's still higher, but not as high, because genetically they're not as similar. Now, the results for ADHD, is your question, are mixed. It looks like the relationship between OCD and Tourette's may account for some of the ADHD increase. But people who have ADHD are no more likely having relatives with Tourette's. So that if you look at ADHD only, their relatives are not likely to have more Tourette's or tic disorders. So that doesn't seem to show, it may be just in the individuals who have the disorder themselves. Okay. And autism, there's very few studies, but there is some evidence of a relationship. However, on the other hand, really, obsessive compulsive disorder is a different story altogether. So we won't talk about those. So you look at Tourette's or chronic tic disorders and you say, okay, a bunch of people with Tourette's or chronic tic disorders, a bunch of controls, what's the rate of obsessive compulsive disorder in the relatives of each? Is it higher? And the answer is yes. There's a higher rate of relatives with obsessive compulsive disorder in relatives of cases compared to relatives of controls. And OCD or obsessive compulsive symptoms, which is likely mild, they don't meet the full diagnostic criteria. And interestingly, if you were in the OCD research realm, not the Tourette's realm, but you're in the OCD realm, and you're interested in learning about what's the familial nature of OCD, relatives of OCD people have a higher rate of tic disorders compared to controls. So both ways. But other studies have shown that there are both shared causes, genetically speaking, and distinct causes, meaning that there are different kinds of maybe OCD, and in particular, OCD in the Tourette's world is more lining things up, getting things just right, where maybe some of the other symptoms of OCD, like hand washing and stuff, is less pronounced. But there is some overlap, but there are some distinctions as well. Okay, so now we've said they are familial. Now let's look at the twin studies to see what proportion might be due to genetic factors. And these twin researchers can be very specific about their numbers, but I don't want to get into that kind of numbering system. Like, 67.75% is due to X. That's too specific that they can do, okay? But basically what these studies do is, if you remember your high school biology, identical twins share approximately, your teacher was wrong, 100% of their genes in common. Identical twins are not exactly 100%. We can talk about that another time, but basically close. And non-identical twins share about 50% of the genes in common. So you have a study design that's sort of pre-made, a natural experiment to see the difference between the two. So what you're looking for, if one twin has it, what's the chance that the co-twin has it? Remember, they're twins, they're two different kinds of twins. We call one kind of twin when they both have it is a concordant twin pair, and when one has it and the other one doesn't, they're discordant twin pair, right? That's just the definition. Now, if the disorder has a genetic contribution, you would expect a higher amount if one has it that the other also has it in identical twins as compared to non-identical twins, because there's more genetic oomph, right? But I already told you that chronic tic disorders and Tourette's is familial, so you would expect that the fraternal twins, the non-identical twins, will also have some concordance, but it's the amount, okay? So, for Tourette's, there's a higher concordance in identical twins than fraternal twins. And if you include these other chronic tic disorders as being affected, so one has Tourette's and one has a chronic motor tic disorder, that concordance goes up even more. So depending on how you define the disorder, if you include both as being affected, the concordance is even higher for identical twins compared to non-identical twins, okay? And they estimate, there's different studies, they estimate the overall heritability at 77%, which is quite high. But I don't want to get into that number, it's really not meaningful at all, okay? But basically, these studies show that there's a genetic component. That's the best way to take home message here. It's familial, and a part of that familial aspect is due to genetics, okay? Now, these are the average overall, because still no DNA is being looked at in these studies. So, in those Tourette's twin studies, they also show a higher concordance in OCD. One twin has Tourette's, the other twin has OCD. Interesting, right? More biology, there's some kind of genetic, more biological looking feel to it. And again, if you were in the research realm of obsessive compulsive disorder, and you get a bunch of twins who have, with one person at least having OCD, you'll find that the other way happens. Person has Tourette, OCD, the other one might have Tourette's or other chronic tic disorder. Unfortunately, there's no really good twin studies for chronic tic disorders versus ADHD and autism. They're just not there. They're hard to do, and they're not going to be around. Okay, so in summary so far, the family of twin studies say that Tourette is familial and has a genetic component, and that these other chronic tic disorders, the motor and the vocal tic disorders, are probably biologically, I hate the word milder, because I've seen very severe chronic motor tic disorders. I don't want to call them milder, but they only have the motor because they don't have the vocal. But they're biologically related. Okay? And it's got to the point where many researchers who are doing genetics work will just combine Tourette's and other chronic tic disorders into any chronic tic disorder. And that's the common research kind of thing we do. So definition of the DSM is not what we think of in the biological sense, and people don't fit those roles. There's also evidence for shared cause for these chronic tic disorders and obsessive compulsive disorder or symptoms. Okay? And the relationship between chronic tic disorders, ADHD, and autism spectrum is complicated. But there is some kind of relationship. We're just not quite sure in a genetic sense. Okay, so that got you through why we think Tourette's, chronic tic disorders have a genetic component. So now it's time to get into the DNA, right? However, not everybody's up on genetics, as we have to focus on. So I want to try to bring you back to that one neuron in your brain that you had in high school biology about genetics. I have to bring you back up to speed a little bit and then bring you forward, because in high school, things have changed a lot since then. So, a little review. First, DNA is like a blueprint for a building, okay? It doesn't build the building, it tells you how to build the building. That's what DNA is. It tells you how to build proteins. The proteins are being coded for, okay? So, you have a blueprint. If it's a good blueprint, great. If you have a change in that blueprint, you're gonna have a problem. Potential problem with the building that's being produced. If it's an error. Same with DNA. Now also it could be a better building, or it could be a building that's 4 stories higher or something. But it also, like DNA, could also improve things. But most of the time it's a negative thing, and it could change. So, from your back in your high school biology, the DNA, the CTA and Gs, remember, they can change. From one person, you might have an A, another person might have a G here, and another person might have a T here. Bases. So I don't want to get into that detail, but that sequence can change between one person and another. In some people, it all depends on where that change is. Is it part of the genome that codes for a protein, which is only 1 to 2%? Is it in a part, does it change the protein drastically? So that's what I want to talk about. So some of these point mutations are silent, meaning you can have change and there's absolutely no difference between one person and the other in that gene. It doesn't do anything. It doesn't change the protein that's being coded for. Blueprint, remember. Then there's changes that does change the protein. Okay? So we call these synonymous. Synonym. Non-synonymous, there's a change. The protein that's being produced is different. Okay? And within those non-synonymous, there are some bad changes, meaning that the protein that's being produced is totally different. It doesn't exist. It does something totally different, right? It codes for something totally different. And for those people who do work in genetics, those include the nonsense mutation, frame shift, and other kinds. I don't want to get into the details of those, but it's easier to call these likely gene disrupting, meaning that it's disrupting the protein that's being coded for. Okay, now something you probably didn't know about if you only took high school biology is the structure of DNA also changes. So if you remember, you get one chromosome from mom and one chromosome from dad, right? So you get one of each from them. And we used to think that only in rare situations that there is big changes where you have an extra piece that may include many different genes that some people have, and it causes really severe disorders. But it turns out we all have sections, structure that's a little different, meaning that there's pieces of the DNA that's either too much, so you can see in this situation, this person has 3 sections of C. I'm not going to get into how big it is or not how many genes it is, because that's part of the problem. How many genes does that piece include? Okay? Or in other people, they have a deletion. They only have 2 copies of the C section. Okay? We all have this. surprisingly enough. So these can change, and we have a definitional change. They call them copy number variants, or CNVs, if they over 1,000 bases are either have too much or too little, and if it's smaller, we call them indels, which we call insertion and deletions. I want to get into the details. That's just the definitional kind of change. So, our understanding of genetics has changed over the last 10, 15, 20 years. We used to think one gene, one disorder. Cystic fibrosis, Tay-Sachs, sickle cell, Huntington's. One gene, maybe different mutations within that gene, but one disorder. Oh, that was so nice back then. It was just one to one. And of course, we're humans, and we have many more genes than every other species out there. Also not true. So, that's not true anymore. You have to get rid of that idea of when you took high school biology that it was one gene, one disorder. We're not in that realm anymore. Those were the low-hanging fruits of finding the causes. So initially we would do these when we thought that was the only cause, one gene, one disorder. We get these big families with many people affected and many people unaffected, and it really worked really well when it was just this one kind of one gene, one disorder. But then we had problems with these other disorders. We couldn't find it. We found one positive finding and nobody else would find it. And it was not working anymore. So we said, okay, well maybe there's a different hypothesis of what causes genetic disorders. And it happened to be more in the common kind of disorder, not like 1 in 3,500. So, a new hypothesis. Common disease, like Tourette's, or chronic tic disorders, common variant, meaning it's really common in the population, meaning that it's like over 5% in the population. And the hypothesis is that each one of these small little variants add together in concert, that if you have enough of them, you will get the disorder. So additive, many different ones. And if you didn't have quite enough, you wouldn't have the disorder. Okay, that would be the hypothesis, and how you would then go about looking at this is the thing called genome-wide association, GWAS. This is, if you look in genetics, you'll see these synchronous GWAS studies, where they would look to see if some people would have, look at 10,000 cases versus 10,000 controls and see what area the genome pops up more likely than others. But remember, it's supposed to be additive. Another possible hypothesis is, again, a common disease, but now a rare variant. Something rare that maybe more, only a few families maybe have that variant in that gene, okay? But if you got that mutation, that variant, you're probably gonna get the disorder. Okay, it's different than that little additive part. You have this really bad hit, you're gonna probably get the disorder, okay? And for those, we would do these sequencing studies. And that's basically reading the code, CTANGs, of at the moment, just the part of the genome that codes for proteins. We're starting to look at the other parts, but we don't really know what to make of that yet. It's sort of an unknown area, but hopefully that's coming down the pipe. So these are two different hypotheses, okay? So currently, the genetic architecture, as we call it, like what is the full idea of what causes Tourette's, is result of a complex and heterogeneous, meaning many genes that involve both these common variants, maybe as a background risk that if you have a really new bad hit or some environmental problem, you will get the disorder. And rare variants, these really bad hits. Okay? They may interact, I'm using the word may because we don't really know for sure, with some environmental causes. Now, when I say that, it's not like, the kid is out playing and having too much sun or something. We're talking about in utero, because this is a neurodevelopmental, neuropsychiatric disorder that it's all occurring in utero. how the neurons are finding each other. So things like smoking during pregnancy, people have looked at conflicting results. Things like that are more of the environment that may interact with it, okay? But these are really what we're focusing on. But I didn't want to give, lose the fact that there's still a potential for environmental causes. So we formed the Tourette International Collaborative, and yes, I came up with that term, and I really love it. And this is our group. And even is growing since then. We have people in the United States, Europe, Israel, and South Korea. We had a group that worked a lot on autism, and they had already formed this really good collaborative group, so we sort of grabbed them along with our Tourette's work. We have a special questionnaire, a self-report, that has been translated into all these languages as part of my work, and making sure it's all done. And all these sites recruit families, collect blood, send it to our university at Rutgers. We process the DNA, and you're sending it out for analysis. One thing about our study that I want to point out is that we are going to be a sharing repository. The idea here is many researchers over the years take their data and hold on to it. We are going to not only do all the sophisticated analyses ourselves, but we will open this up to any qualified researcher from around the world. So if somebody comes up with a new fancy schmancy new data analysis, come on in. We want more research. We need the field to move forward. In the autism world, they have a lot more money and they have a lot more researchers. We need more researchers to move this field forward. So one thing I just want to point out is that's what we do. It will be a sharing repository. And it's not us who decides who gets it, it's the National Institute of Health who decides. So even if it was our competitors, they could get it, right? So the idea here by our group is to further understand the genetic architecture of these chronic tic disorders, and it will be a sharing repository. We're going to get a large sample, well characterized, meaning who has what in the family, use all the state-of-the-art genomic technologies to understand the cause of what genes are involved, and we're focusing on those rare variants I talked about before. They're common disease, rare variant kind of studies. So we are looking at the sequencing of the DNA, and we're also looking at the structure of the DNA, see if there's anything different. So we are focusing on these rare de novo new mutations, and I'm going to explain to you why. When we sequence people, even just the small part of the genome that codes for proteins, we're going to get many, over 50,000 variants for each person. Obviously, those are not all causal for the disorder of interest. How do we filter out all of that stuff that's not important and focus on the ones that we're interested in? We need a way, a study design that will help us get rid of those other ones and focus on the ones that really potentially could be causing, okay? So there's two ways mainly, family-based and de novo, and I'm going to talk about them. And then once you have looked at those filtering, you'll say, okay, well this mutation is synonymous, we talked about before. Doesn't change anything. That's not going to be causing the Tourette's. So we're going to get rid of those. And now we look at the fewer that could potentially be causing disorder and try to understand those. So, the two methods is you take these big families where many people are affected, many people are unaffected, and you try to trace through the family areas of the genome that seem to go along with the disorder, called co-segregation. This worked really well when there was one gene and one disorder. Doesn't work so well anymore. But we have a bunch of these families with Tourette's, chronic tic disorder. Many people affected. But we've now focused on, and what the world has in autism and epilepsy has done, is start looking at a new study design in which we look for families that we call them simplex trios, where only the child is affected. okay? And what we're looking for, is we're trying to catch that one moment in generations when a new mutation has occurred, and then we can compare what's different in the kid versus the parents. If there's something different between them, all that other stuff that's not important will go away because both parents have that. So we can get rid of that, and we only focus on the things that are new. So new mutations are rare. New mutations in the genome, part of the genome that codes for proteins, is even rarer. And bad hits in those things that really change the protein are even rarer. So rare times rare times rare times up, okay? And so we're going to be looking at the sequence changes that we talked about, and we're going to be looking at structural changes. So here's some of our families. Darkened in symbol is affected with Tourette's. Gray symbol was a chronic tic disorder, which at the time we didn't know how to handle. And then we have some others that we just had some kind of odd tic thing in the history that we weren't sure about. So we have those sort of hatched kinds, right? We have a few of these, quite a few of them. But they don't really, so here's the first kid who came in. His sibling has chronic tic disorder, and his other sibling does not. Mom had a Tourette's, uncle had Tourette's, first cousin had Tourette's, grandpa had a chronic tic disorder, okay? That's in case you don't pay attention to pedigrees and stuff like that, which I see all the time. So we have a lot of these kind of families. But in this new study design, which has been very successful in understanding these complex traits and disorders, it's been estimated that there's only one new mutation per generation that changes the protein bad. It's sort of a bad hit, like likely gene disrupting. So rare, one per generation, right? And does that, we're looking for those, right? And this has been really helpful in understanding other disorders like epilepsy, intellectual disability. This is study design, they're very common, and autism, very common, okay? We reliably and systematically helps you to identify these rare mutations. So let me give you pictorially what this is. We're trying to find families in which there is only one person affected, the kid typically, right? And the idea here is to enrich our sample. We know that there's some that are not necessarily new mutations, but it's trying to enrich our sample for these new mutation kinds of families, okay? So we have a bunch of the affected kids, a bunch of parents with not being affected. And what we do is we use the sequencers to look at the genome, the exome, which is part of the part of the genome that goes for proteins, and we're looking for structural variants. Okay? So conceptually, if the idea of the genetic architecture of Tourette's is due to new bad hits, that if new bad hits can cause these disorders, you would expect a higher amount of these new bad hits in the cases compared to controls. Just because that's many different genes, right? Many different places, but you would expect more of them. Burden, that's called a burden allowance. So higher rate of the likely gene disrupting, new, remember new, okay? That's the burden analysis. Then, over time, because if you think there's many different genes, over time, at some point you're going to get a second family with a new mutation that's really bad in the same gene. But maybe a different mutation, but it's the same gene. Wow, that's noticeable. If two different families have a new mutation in the same mutation that really disrupts the protein, that's pretty notable. Okay, so that's sort of the lightning striking twice concept. So in 2017 and 2018, we had two publications using this study design, the simplex trio's new mutations. And for those of you who need to know more details, in the methods part, we sequenced and genotyped 777 trios. That's times 3. That means 1 kid and 2 parents. We did that for each one. We compared them to data from a control population that used the same sequencers, same kinds of analysis, same kind of molecular studies that were being done. So there was no, we call batch effects. Okay? And what they were so Simon Simplex is a huge, giant autism study that uses somewhat similar kind of methods that we are using, but they've been around longer and they come with a very large amount of money. We're using the unaffected sibling in that. So they had both parents, they had a kid with autism, and they had an unaffected kid. We're using the unaffected person in the family as our control group, our comparison group. Okay? And we're looking for these burden analysis. Are there more new bad mutations in our cases compared to those controls? And are there any recurrent hits? Same gene, different, maybe a different mutation in the same gene that causes this disorder. We're looking to see specifically if there's something that really disrupts that protein. And we're also looking for these new structural variants. New pieces of DNA that's been deleted or added. We talked about those structural. Okay, so what we found, this is a summary from both studies together, 2018 paper, compared to controls. The cases had a significantly higher number of these new damaging mutations in the sequence variants. And for those who were statistically predicted and all that, there's a relative risk and the p-value, but I'm not going to get into that. But it's significant. So that there was a higher amount of that sequence variance in our cases compared to controls. Yes? There was also a higher amount of those structural variants. Pieces of DNA. Could be large pieces, could be small pieces, that are either added or deleted. Okay? Compared to controls. And interestingly, the genes that were found to have the new mutations in these, some of them were found in the OCD world research. They've been associated with OCD. And the structural variants, the genes that are within those sections of the DNA that's been pulled out or added, have been associated with autism spectrum disorder. So it all comes together back again with what we were talking about with those family studies and all that. So burden analysis, check mark. We also, just based on the number of people we had and number of families, we estimated there is probably about or over 400 genes that each can cause Tourette's. So drop the idea of cystic fibrosis, Tay-Sachs, sickle cell, one gene, one disorder. You have to think of that world of how these genes could potentially come together. And about 10 to 12% of the cases are due to a new mutation. So some families keep coming up to us saying, nobody else in my family had it. Yeah, well, that's because there was a new mutation. However, even though they have a new mutation in that kid, remember it was this very specific moment in time. When that kid gets to be an adult, they still could pass on that new mutation. It's important to understand that. We're doing a very specific moment in time, but then we'll go back to generation to generation. So before we get to the point where the next generation starts having symptoms, we want to get a better handle on this and get better understanding so we can maybe come up with better treatments. Because I know that you wanted to find a better treatment compared to what that were created for other disorders, not Tourette's. And if we have a better understanding of the network that you were talking about, we would have maybe a better treatment. And this is where it's coming from. So, lightning striking twice. All of a sudden, after a certain number of them, we started finding some genes that in one family that have a new mutation, and another family have a new mutation in that same gene. Very notable. This is what happened in the autism world. They started doing this, and they started adding more and more genes that were being hit more and more. Okay, so these are the genes which don't mean anything to you, but don't worry about it. All I have to say that the cells are 3-1. We've even found some others, heard of other new mutations in other families. So this is really excited about this one. So when you look at these, they don't know, even when we look at them, we don't necessarily know how they fit together. Remember, we have to think about how these hundreds of genes can fit together in a pathway or a network in the brain that's getting disrupted. So there are these databases that you can say, okay, here's a set of genes that we're interested in. How do they fit together? Do they fit together in a certain kind of proteins that are involved with something? Okay? So you throw these into a database. And they come back with, okay, these genes seem to have, are involved with something. With the protein that's being produced is something involved with these things. Are they totally disparate, or do they have some commonality? When we did this, we found that a number of our genes are involved with what they call cell polarity. And I know that doesn't mean a lot to you, and it didn't mean a lot to me because I'm not a neuroscientist. But the idea here is cells need to have a front and a back, polar, okay? During development, those neurons need to come together in the brain. You know how many neurons there are. They need to find their partners. If they don't have a good top and a bottom, they may not fit together in that network really well that we heard earlier. They may be off a little bit. They don't connect to the right partner. Okay? So but if you have a problem with your front and back, you're going to potentially have a problem with getting to the right place. So, affecting the correct location and making the right connections. Obviously, this is still new. Who knows when we have our next batch if that'll still be the case. But obviously, when it came out of the database, we're like, oh, this makes total sense. Okay, so here's sort of the pictorial aspect of our results. In our sequencing, we found a higher amount of our simplex trios compared to our controls. We found a higher amount of structural variance in cases compared to controls. We have a number of high confidence genes, and the CELSR3 is our one we're really interested in. We found that these genes are involved with cell polarity, and the sequence variants overlap with genes that have been found in the OCD world, and our structural variants overlap some with the autism world. Some distinct, some together. So what's next? Well, we need to recruit and analyze more families. This design is working, but we need many more. That's why you have such a large group of tick genetics, to get as many people as possible. We got a new grant, 2018, to continue this work, to get more families. And then we need to now look at what do these genes do, either with or without the mutation. Why is it causing these movements? Or neuropsychiatric, OCD, and ADHD. What's happening with those neurons? And for that, we do functional studies. Okay? So that's what's the next sort of step in our process. So what are functional studies? We need to understand why there's a what this mutation does, and why does it cause the symptoms. They're called genotype versus phenotype. Phenotype is what it looks like, and genotype is the gene and the mutation. Okay? And we need to determine if this mutation really does cause the disorder. And there have been a lot of functional studies been done from any kinds of studies. I am not going to review all of these, like animal models, mouse models, with the mutation. There's all kinds of others. Looking at the transcript, that's how much messenger RNA, for those people who know anything about genetics, is there. How much of the protein differences there are. Okay? I'm not going to talk about that. I'm going to talk about some of the more interesting science fiction-y kind of analysis. So, but it isn't science fiction. So, induced pluripotent stem cells. So you may have heard of stem cells, which are cells that can become other kinds of cells. It could become a neuron, it could become a white blood cell, it could become a kidney cell, a heart cell. Those are what a stem cell, why they're such an important, they can become anything. So you go from one cell to other cells, like stem cells that could become almost anything, and then you may have a mature cell type. Let's call that a white blood cell. Okay? White blood cell. I have enough problems getting blood from kids. You can imagine how because I'm not interested in the white blood cells. I'm not interested in what's in the blood. I'm interested in what's happening in the brain. Right? That's where it's all happening. Well, obviously, I am not going to go out and say, hey, can I have a neuron? Right? So, but that's what we're interested in. So let's say we have a white blood cell. Well, what if I can, using certain chemicals and various things, turn that white blood cell back to a cell that can become almost anything? Wouldn't that be cool? Well, that's really what we're doing. We can easily now, it's a lab technique, where you can take a white blood cell or a skin cell, turn it back to a stem cell, and say, okay, now become a neuron. So now I get neurons from people who have a certain disorder. Interesting, right?
That's wild.
You can do it for all kinds of cells. Oh, they can become big, those stem cells can be anything. Okay? So in 2006, they came up with this. 2012, they won the Nobel Prize because it's such an important technique. Okay. So the idea of doing this is to say, I'm going to take that white blood cell out of somebody who I know has a very interesting mutation, like that cells are free. I'm going to make that white blood cell become a stem cell, and then make that cell become a neuron. How does that neuron look different? Does it fire differently? Does it have maybe fewer branches? Is it supposed to make connections? Does it have electricity, the electrophysiology, which is like, does it fire the same amount and all that kind of stuff? We're going to look and see how those things are doing. But now we can do that with that technique. We can have these individuals, we know that they have a very interesting mutation. We're going to take that, make that white blood cell back to a neuron and bring it to a neuron. So here, for example, is a neuron that used to be a white blood cell, firing spontaneously like any other neuron. So the problem is, who would be the right comparison? I have a neuron from you with an interesting mutation. Who do I compare that with? How does the branching or the electricity, electrophysiology change? Who do I compare you to? Do I compare you to your mom, to your dad? Remember, only 50% of the genes are in common. That's not the best comparison because there's a lot other things that could cause changes. Right? Really what we want is you with that mutation and you with everything else without that mutation. That would be a really great experiment, right? That's what we want. Well, we want these things called what we call isogenic controls, the identical person without the mutation. How do we do that? Well, you may have heard in the news, don't come yelling, a thing called CRISPR. There was a guy in China that came up with this thing where he changed the genes for a couple of kids for this certain genes, and he got into a lot of trouble. But we're not doing any of that. What we're doing is going in, taking the white blood cell, bringing it back to a stem cell. We know the mutation. We can, in those white blood cells that became neurons or stem cells, change back, fix that mutation in you, and now say, both of them become neurons. Now I can compare people with the neuron with the mutation, and the neuron without the mutation, and everything else should be the same. How are those two neurons different? Right? You understand what I'm saying here? So that's what we're doing. So I'm not going to get into the details of what CRISPR is, but again, it's becoming a lab technique. It's really fascinating. It was really big news, and people were fighting over who did it first and all that. I'm not involved with that. But we can now create differences. The same person without the mutation. Okay? So in summary, the genetic epidemiology studies answer a certain set of questions. Is it familial? Assuming that it is familial, what proportion is due to genetic factors? And then, where are those genes located? Our idea of genetics has changed over the years. We used to think one gene, one disorder. Now, many different genes can cause certain disorders. So different, what's different in you, what's one in you is different from a different person, different from a different person. Okay? Hundreds of genes. These sequencing studies does cause a huge number of data. It's a big data problem. For anybody who's in statistics, we need to find ways of filtering out those non-important ones. And that's where we use those, either those big families or these new simplex trios. And we are, in our group, are looking for these de novo mutations in these people who seem to, in the family, look like they're the only ones affected, simplex trios. And we're looking at taking those genes that we're finding and putting it into functional studies to see how they're fitting together in the network analysis I mentioned, and then looking at functional studies to see what's happening in the bench, looking at to see what's different, what's happening. So, I started off with people who were pretty nice. I assume many of you know Tim Howard, goalie. We also mentioned goalies here, but in Canada we think of different goalies. When you think of different goalies than maybe the rest of the world, you know. But we're not talking about hockey. But Tim Howard, he just retired as the goalie for the Colorado Rockies, Raptors. But he was the goalie for the U.S. soccer team for a number of years. And the World Cup in 2014, he had 16 saves, which means that the rest of the team was not very good because they had so many people hitting, but he did stop a lot of them, but that's good. He has Tourette's. He was born right near where our Heiman University is. He's very close with our group, the New Jersey Center for Tourette Syndrome. And he has been very instrumental in trying to bring the positive aspect of things. I wanted to talk to you a little bit about that. So we created with our group, this other group, the Tim Howard Leadership Academy, and a number of Canadians have come down and been part of this, where for like 4 days they will come and live in the dorm at Rutgers. They're like older teenagers, and we have people who are adults who've had Tourette's, and it's a way of learning about the disorder, learning about self-advocacy, and learning about, all the other aspects, relationships, friends, all that kind of stuff during those really hard times. And it came up with this one thing, be one of the greats with TS. I think that was just such a cool thing. Right?
Right.
So, I want you to hear a little bit about Tim. Hopefully this is going to work now.
It took me a while.
I was 9 years old. 9 or 10 years old is when I finally got the diagnosis. I think Tourette syndrome has played a positive role in my life, and people sometimes are surprised when I say that. It's hard to describe how I've channeled my TS. There's tough days. I've just come to cope with it and to, most importantly, learn how it affects me. But I also feel like I want to be great. I strive for greatness every day. I try and be a great father. I try and be a great soccer player. Achieve greatness through playing and hopefully winning a lot. My mom's been amazing. She created a life for me and my brother that nobody could have. Her hard work, her tenacity, her ability to keep us grounded and focused. I think you always need positive people around you. That's one thing I try and preach to families who are affected by TS is there has to be love and positivity and encouragement, because self-esteem is the best medicine. There's such a misunderstanding in the general public about TS. It's been sensationalized by Hollywood, it's been ignored by educators and doctors, and it's real, it's there. We have to cope with it and deal with it every day. And one thing that struck a chord with me right off the bat with the grades was that it was bold, it was in your face, it was a statement. It was basically like, look, get on board or get left behind. Like, this is who we are, this is what we're about. It's okay, it's cool, and the sooner everyone gets on board and pulls in the same direction, I think the better off the TS community will be.
So, staying with the grades. So here's their manifesto. I'll leave it up for a second for you all to read it. And this is online, by the way. So, as you can see, he's a really great role model. They have this academy they have every year at Rutgers, where the kids come in and last year he did a video chat with them, and they were like, I became a goalie because of you. You know, it was really touching. And the kids really get something out of it. And it is open to anybody worldwide, this academy. So I would like to acknowledge all of our collaborators from all over the world. A lot of them in different languages. I get to visit some of them, which is really cool. And if anybody works in the science, you'll be reading some articles, you'll see some of the names up here.
That's right.
So I also like to acknowledge our funding agencies, the National Institute of Health and Mental Health, and especially the National Institute of Mental Health. Faith Rice, who is the head of the NJCTS group. Tim Howard, and I love this picture because look at the contact that they have by eyes here. They can see that they're making real contact. And there I am, just sort of handing out books. That was my job today. And also the families who got involved in helping us really move the field forward. The NJCTS had one of their kids with Tourette's created a cartoon here, which I had a couple of copies here. You can order them online, I think on Amazon, or you can go to the njcts.org. On Twitcher, which is a cute little thing. So she's got superpowers, which is when she twitches. So I want to thank you all for this. As I mentioned before, the Tourette Canada table, which is just outside, there's a number of brochures about the Tim Howard Academy, about NJCTS, which has an archive of webinars. So you can sit down at any time, late at night, weekend, and just listen to information. So if you're alone and you can't get to the Tourette Canada people easily, there's more information out there for you. Feel free to use them. It's not just for New Jersey families, okay? Thank you. I did it quickly. I know there was a lot there, but I think I have like 15 minutes to do questions. So I think I'm going to open it up for some questions for people. Yes?
When is the camp?
We have a camp and we have the Tim Howard Academy. Camp is like early, like in June, where we have they come every year to this old Y. I don't know if you have Y camps here. Do they have a Y camp? It's like tater tots and stuff, and they have cabins with kids. The families stay together. The whole family goes.
That's the 4-day intensive?
That's just like a little weekend thing, and just kids getting to be themselves, basically. Having fun, and you have a community of people that get to know each other. The Tim Howard Academy is like in August, and you have to apply, you have to get into, make sure that that the kid can make to do the stuff. So families we had somebody come from Australia 2 years ago. Parents came, they dropped the kid off, and they spent the weekend in New York City. So that's what that is. But they say they get so much out of it. They do relaxation techniques, they do all kinds of stuff. Other questions? Yes?
You may have answered this, so forgive me if you did. I was trying to follow along with biology.
I know there's a lot of stuff there.
But it was very interesting, thank you. My question is, if one child has it, is there an approximate percentage, chance that the other sibling will get it?
I was wondering if somebody would ask me that question.
Okay.
So I currently have just submitted a publication to ask this question. Now remember, as I wrote and I put in one of the slides, that this is an average of all families. Those family studies could be used where they just look at how many cases, what's the rate and the relative compared to the controls. We can use that data to get you a overall average estimate of what's the percent of somebody else getting it. And my paper is in review, but I figured somebody would ask that question. So, let me just put this one out there. We are looking for families. We're looking for more families. There are brochures out there, or you can email us here, in which we're really looking for those families where at least only one person has the chronic tic disorder, okay? And I mentioned the webinars, okay? So, here's the numbers. So, if one person has the disorder, there's about a 30% chance that a first degree relative, okay? So, if one kid has it, there's about a 30% chance another kid might have it. Or if the father has it, what's the chance the child will have it? It's about, from just some of those family studies, average overall, there's nothing about you and your family, okay? Just want to make sure those are the numbers we have from that. And then in the second degree relatives, it's lower, it's still higher than the general population. And I guess before I answer that question, I want to just get, because I want to make sure what you understand what this means. So I want to clarify that obviously chronic tic disorders are highly heritable. And these are data from family studies. It's an average across all families, not your specific family, okay? It is due to a complex inheritance of many different genes, and they may interact with environmental factors, and there is currently no test available. If you remember, possibly 400 genes. How can I test that without even knowing all the different genes yet? We can't, there's no test available. If somebody asks for a test, they're probably going to be getting you a wrong test. It's probably not the one to do with in your family, okay? 10 to 12% are new mutations, but these new mutations can be passed on to the next generation. Okay, just make sure you understand that. And the idea of severity, which is really what most people really want to know. If I have one really severe kid, is that next kid going to be really severe? It doesn't seem to be true. We see many different families where one person is really severe, they came to the attention of the doctors because they have severe, but we look at all the parents and everybody else and we're like, Dad's ticking right in front of us, and they all say, no, nobody else is ticks. The mom says, you're doing it right now. We commonly say this, but it's just a small thing that never bothers them that much. So it doesn't, so we can't say anything about severity. And of course, severity changes by the day, by the week, by the month, by the season, by the year, right? And over different years. So the quality of life varies between different people. Okay? So I wanted to make sure I say that because it's really, make sure you understand that this is nothing to do, it's an average overall.
Thank you.
Yes? You want?
I was just gonna make the point that last night, my son said, like, Mom, when I get big, what's my chance of?
I'm sorry, I have to speak up.
What is our children's chance of passing it on to their children? They're asking, but essentially you answered
Yes.
30%, but we don't know.
We don't know much more than that. Until we have more information about the genes, we don't know. And we need to get to know more about this until the next generation. That's why I'm asking people, if you fit to that criteria, consider participating, because we need to move that field forward before that next generation gets there. And also, just to let the public know that this is a real disorder. It's not the TV version of Tourette's. And also the medical professionals need to know this so that people can get diagnosed quicker. To understand that it's not just the swearing disease that some doctors, that's what they learned in medical school. It's more, there's more to it. You have a question?
Yes, I do. I just wanted to know if any of the research that you're doing relates to the autoimmune system.
No, not yet.
Not yet, that's coming.
Been a lot of questions about this autoimmune stuff.
Yeah.
And a lot of controversy about this autoimmune stuff.
Well, it's only
The
because
pandas and stuff.
when you talk to people, it's not just, I don't know about anybody else in here, but from friends of mine from home, if you have one child, the next, oh no. Can you not hear me over there?
I can hear you.
Hardly. You can't?
Can't hear you.
The autoimmune system, with a second child that maybe has diabetes type 1,
Mm.
or autism or Asperger's.
Right.
So you've got that, but diabetes is.
So the autoimmune is different than like a neurodevelopmental disorder.
Yeah.
We are interested in individuals, we do see a lot of autoimmune and allergies in individuals with Tourette's, whether or not that's causative or something that's going along with it, because that's whatever's changed, it's changing something with autoimmune, we don't know. But the idea of that it's causative, like this thing called pandas that were big controversy with PAMs and all that kind of big controversy, and then maybe not.
So
Yeah. Way in the back.
How do you participate in this study from Canada?
So we have a blood company that will come to people. So we do everything, but we can do it by mail. We would send you the questionnaires, and it's a bit of a long questionnaire, as you can imagine. Everybody's tics are a little different. Everybody's OC is a little different. So we have to have a big wide net of questions. A lot of people say, I learned a lot about myself or learned a lot about my kids. Because if you don't ask the kid, hey, you can see the tics. But if you don't see the OC and you ask them these questions, it's like they're like, wow, I didn't realize that this is what they were thinking. Okay? So we need to do that by mail. And then we have Dr. Robert King is at Yale, he's the medical director of Yale's Child Studies Center on Tourette's and OCD, is very well known. He comes down to Rutgers once a month and does the evaluations for us. He's a great guy to chat with. And for bloods, we have a company that I'm told that can go to Canada. I don't know all of Canada, but we would see what we can do. I don't know how easy it is to get a blood sample drawn through Canada, but we would see if we can arrange it, because we need families. Yes?
My daughter has a cousin with Tourette's, but no one in our immediate family. Would you still want her to participate in the survey?
Sure.
Yeah.
It's one of those situations where it's a relatively common disorder, and then we do see some first cousins, but they could be coming in from something else totally different. Yes? Any other questions? Okay, thank you very much.
I want to thank Dr. Heiman for his excellent presentation, and I just realized that we have had a very rare privilege of having someone that is doing the research that we hear about. We've had that shared with us. You've opened up a window of something that is always sort of mysterious. How is it done? How do you come up with the, how do you do the research? This is a big study, and I so appreciate you coming up to Winnipeg to share this. And also, I know you've been meeting with families and you've been participating. It is a privilege that most people would rarely have, so I want to thank you for that. And I also want to thank you for the work that you are doing and your team is doing, because this genetic work is to benefit our children, our families. And so as you heard Dr. Heiman's talking about this, it is to find and to isolate so that new treatments, better treatments can be found, and working together. Yes, so
it goes very along with our last talk.
Very much coordinated.
So thank you so much, and we really appreciate the work that you're doing. And I also want to add that Faith Rice, who works with Gary, is extremely generous too, and she has offered all of her research with Tourette Canada, and she and Jenny, our executive director, have been in contact and sends up information to Tourette Canada. Extremely generous. As she says, it is all about our kids.
She believes it.